Excellent. All right. So, as I was saying, for those of you who do not know me, my name is Chad Johnson. I'm the optical microscopist at the bioimaging platform. So I run these workshops throughout the year. So I do part 1 and part 2 of QuPath, as well as 4 parts of Fiji. I've cut the workshops down from the longer versions. This workshop used to be 4 hours. So based on feedback, I've cut it down to 2-hour sessions, which makes it a little bit more palatable. But I will be running the part 2 for this later on in the year, probably about October. And I've started the recording. Yes, good. Okay, so as I said, I'm part of the bioimaging platform. So we handle Latrobe's optical EM and histology and flow cytometry capabilities. So if you need any assistance or just some advice,
signed up for this
bioimaging at latrobe.edu.au.
What's it for?
If you'd like to, we're on fairly active on social media, especially on Twitter. I've moved to Blue Sky for obvious reasons, but still maintain my Twitter platform. We're also on LinkedIn, and I've just started a YouTube channel, which I'll be posting when I actually have the time. It's posting questions, sort of like Fiji techniques, image J techniques, QuPath techniques, microscopy techniques, that sort of thing. I'll slowly sort of put those up. I tend to run these workshops via cinema rules. So if you can please turn off your microphones and cameras, which I will be doing, at least for my camera, I'll be turning off just so we don't distract people and so we don't hear background noise, etc., etc. If you need to ask a quick question, I'll be keeping an eye on the chat. And oh, it's all right, Christine. I was getting to this anyway. We're in early days, so yeah. If you do need to ask a question, I'll be keeping an eye on the chat, and just type it in there. If you have a larger question that's going to take a bit more sort of going over about specific projects and things like that, feel free to ask to email me afterwards. And most of you know where my office is anyway, so feel free to drop by and have a chat. So I'm going to turn off my camera. Okay, so the aims of this workshop is to learn the basic functionalities of QuPath, to use the software to automatically detect and classify cells in histologically stained tissue, and to get an idea of the vast amount of processing and analysis capabilities that the software has to offer. Now, a lot of software that we use, we usually barely scratch the surface. For Image J is a good example of that. Fiji has so many functions that most of us will just never even touch. There'll be functions in there. I've been using Fiji for 15 years now, and there's probably buttons that I have never even needed to use because of they just don't really apply to what I'm doing. QuPath is no exception. There's a lot of functionality in there, and there's usually something for most people if you need to do some sort of work within image analysis space. Now, in that regards, use your imagination. So we're going to go over some fairly specific examples that do tend to show the broad applications of the software, but it's not limited to just that. There are lots and lots of things that QuPath can do that you can either use specifically or bend to what it may not have actually been designed in purpose for in the first place. So you can do all sorts of stuff. This software is mainly for histologically stained tissue. I've used it to analyze things like bee pollen and all sorts of things. So it's not just for these specific examples that we're going to be going over today. So a basic overview, we'll be going over introduction to what actually QuPath is, what the basics of the functionality are, selecting using ROIs, opening images, things like that. Channel navigation is a big part of using QuPath, being able to access different channels, optimize different channels. We'll be going over that. And then we'll be going over some basic analysis and cell detection. So this will form a foundation for the second part, when we will be using what we know to then move into the machine learning algorithm. So training QuPath to detect specific objects and specific cells and things like that, that are a bit beyond what we're going to be sort of doing here. So previously, as I said before, we did do that as part of this one workshop, but I just based on some feedback, I've broken it up into two parts to make it a little bit more bite-sized. So introduction to QuPath. So what is QuPath? So QuPath is free, which is great. So for us, where software can cost tens of thousands of dollars, having a free, powerful software is incredible. It's open source, which means if you were so inclined, you could completely rewrite it to your own bespoke requirements. It's basically mainly used for slide scanned images, and we'll go into what that is and why we would do it shortly. And it provides researchers and pathologists with a user-friendly interface for analyzing large digital slides. User-friendly software is very, very important, because if it's too hard, then a lot of people just completely avoid it. But QuPath has been designed to be very, very user-friendly. It contains automatic cell detection, tissue classification, machine learning tools for image analysis, and just like any good software, it supports a wide range of different imaging formats, because every time they bring out a new microscope, there always seems to be a different imaging format. And because it uses the bio formats and Amiro functions, it can pretty much just open anything, which is great. It was created by Dr. Pete Bankshead, who's a Scottish pathology researcher. And now it's maintained by an entire community. So he wrote the original software, and it's now basically like Fiji, and a lot of these open source software is actually a big community, because the project just gets too big for one person, and they contribute to its ongoing development. And it's a great alternative to the very, very expensive softwares such as Halo, which is insanely expensive, and VisioFarm, which I've never used, but I hear is equally pricey. This is the original QuPath paper. So QuPath is relatively new, less than 10 years, but that's the original paper if you want to have a look through. This slide deck will be available as a PDF afterwards, along with the recording, by the way. The QuPath website is here, so a lot of detail about the functionality is there, including a wiki and all that sort of thing. So for every functionality, all the functionality, there are explanations and how-to guides here anyway. But Pete's YouTube channel, so Pete Bankshead, he's got a YouTube channel where he actually goes through a lot of the functionality via these instructional videos. So that's a really good resource to have. But there are equally many other people on YouTube who actually do QuPath tutorials as well. So it's a well-used software with lots of resources. So why would we use QuPath? So QuPath can open and analyze most standard slide scanners. So we have a Zeiss AxioScan 7, and it just runs through those files really well, as well as the Olympus VS200 and the Leica Aperia. So those are the three main sort of slide scanning microscopes that you will find, but it's not limited to just those. Now, ImageJ is really good for handling 2D images. It has some 3D functionality, but its bread and butter is pretty much 2D. And its image analysis is, for a free software, it's incredible. However, some data files, especially very large ones, are beyond its capabilities. Now, we're going to be using what's called a pyramidal image format, which is what most slide scanners use. Now, Fiji cannot open these files. They are just far too massive for it to actually deal with. Maybe if it was a small image file, like a very small section, but for the sort of, if you're scanning an entire brain or something, you have no hope opening them in Fiji. So QuPath will change the displayed resolution depending on the zoom level. So essentially when you're really, really zoomed in, it will only show you that particular part of the tissue. And then when you zoom out, it will show you the whole thing, but at a very, very low resolution, which allows the basic laptops to display the data very easily. So I can run QuPath quite happily on my very, very basic Latrobe laptop. So a pyramidal image is exactly how it sounds. It is essentially, you can visualize it as a pyramid shape. So at the very, very bottom of the pyramid, you have the very, very, very large, high resolution images, but layered on top of this are several layers of lower sampled down images. So you might have imaged the original data at 40 times. Then the next layer up will be usually a quarter of that, then a quarter of that, and then a quarter of that, and so on and so on. And if you actually look at the metadata, you can actually see the pyramid. So one would be the full resolution, then you've got half, quarter, 1/8, 1/16, 1/32. So that allows us to essentially peel those layers off like you wouldn't even notice it when you're sort of scanning through it, to essentially zoom in and out of a image. And so when you try to open these up in Fiji, you get this, where essentially all of the layers that are available are given to you as possibilities, and you can choose them. Now, the top layer will be the full resolution, and Fiji would just fall over. Really would not be able to open up. You might be able to open up layer 2, but most of the time we are usually stuck with layer 3 or 4 because that's the resolutions that we can sort of really deal with. But these sort of really large resolutions, this one's not so terrible. I could probably quite happily open up layer 2 because it's only about 3,000 by 3,000 pixels. So it's Google Maps rules when it comes down to it. So if we are looking at Google Maps and we look at Melbourne, we can see Melbourne as a whole. Now, I am in Bundoora at the moment. Now, if I took a screenshot of this map at this resolution and then zoomed in on it, it would appear very, very, very distorted because it's screen resolution. But from this level, it looks fine. We just can't see any of the minor detail from within these sort of spaces. Now, if we zoom in with Google Maps rules, actually zoom in using the magnification tool, essentially what we get is the full resolution. Now, what Google Maps is showing me is not all of Melbourne at this resolution, but just this little subset. Everything else we don't see. And that's how it's able to show us really high detail by peeling off the layers as we go until we get down to a very small area of high resolution. That's just essentially the same thing over again. It's usually a memory issue with sort of so ImageJ can't really, even if we have a computer with a lot of memory, ImageJ just can't really access it very, very well. So we have to really downsample them in order to open them, which essentially leads to undesirable artifacts. So that's QuPath in a nutshell. So now we'll go over some QuPath basics. Now, so QuPath is just released as of last week, version 0.6.0. Previously, we had 0.5.0. I'm still navigating 0.6. It looks almost the same. There's a lot of functionality that they've added, but the general functionality between 0.5 and 0.6 should be basically the same if you have 0.5 already installed. And installing it is very, very simple. You download either the Windows or Mac, and you install it how you usually would for a Mac or a PC software. It isn't a signed application. So you will probably be asked, the computer will typically ask permission because it's not signed, it's not a Microsoft signed application to say, is it okay to install this? It's fine. I've never had any Trojan horses or anything like that because of this. And then to open QuPath, you just double click on it and you will get the QuPath will pop up. Now I'm going to show you my QuPath. I'm just going to jump over to the other screen. This is why I like Zoom rather than Teams, as I can select. I'm sure you can probably do it in Teams. I just don't know how to do it, but I do know how to do it in Zoom. So this is QuPath open. Now, you may have noticed when you opened or installed QuPath that you got two icons. One was QuPath and one was QuPath console. Now, console is essentially gives you a command line as well as the software. Now, you can open either one. I find the console fairly useless because I don't need to delve that deep, but you can essentially run different functionalities from the command line if you know DOS or something like that, or the DOS commands within QuPath. I'd stick with the non-console version. There's really no reason, I think, at this level to go into to delve into that. But either way, you can open both. You'll just get an extra window if you open the console version. Tooltips. QuPath is really, really good with tooltips. So anything that you hover over, you'll get a little bubble. Zoom in my little screen. So you'll essentially get a little bubble that shows you what the tool actually does. And it'll even give you a little, in brackets there, the shortcut. So if I press Shift F, it'll toggle the fill function off. Any of these sort of functionalities should have its own keyboard shortcut. So if you're not sure about a function, you can actually just hover over it and it'll actually tell you what the button does, which is great. Opening an image, you can literally just drag, just the same as Fiji, because QuPath was actually built using Fiji, essentially. You can just literally drag and drop an image. So I'm just going to go to, where did I put my QuPath stuff? There we go. Workshop. That's not it. Where did I put it? Lost it. It's a desktop. Where did I put it? There it is, QuPath Workshop. Okay, so you should have the same data as I have. So if you want to open an image, all you have to do is just grab it and drop it into QuPath and it'll open it quite happily. It might take a little while. There we go. And then it'll open it in the background. It'll ask you what type of images, and we'll get into this a bit later, but essentially it should automatically select it based on what it knows it to be, what it knows from the channels and everything. And you click apply, and then you can literally just use QuPath straight away. So zooming in, zooming out, that sort of thing. So you can just open an image as you please. However, I do suggest if you are doing analysis work to use what's called a project. So project allows you to keep all of your images and all of your data organized in one place. So you can open up multiple images within, you can only open up one image at a time if you drag and drop, but if you use a project, you can open up multiple images, do multiple analysis workflows within the same project and keep everything nice and organized. So up the top, you'll find Create Project. If I click Create Project, I can choose a spot for it. I'm going to choose desktop. I should have Workshop, QuPath Workshop, and I'm just going to create a folder in here, Workshop Project. Now the project has to be in its own little blank folder, otherwise it gets confused. So just select the folder and it will create, no, I don't want to save changes to this image. I'm going to create a whole new folder. And now I have my QuPath workshop, my workshop project. So now I can drag and drop images into here as I please. The analysis pane, this little thing that looks like a ruler, that toggles the analysis pane on and off. If you want to get a bigger view of your image, you can turn that on and off. So adding images to our project. So I can go into add images button right here. And it's going to ask me to drag and drop any images that I'm going to use. So I'm going to go back into my little folder, which I had before. Okay, so there's my folder, workshop data, and I'm just going to drag all of these images straight into here. And they're all ready to add to the project. Now, essentially what it's going to do, it's going to remember the link, the URL, essentially, or the folder file path that your images were. So if you do move the images somewhere else, it's going to when you open your project back up and the images aren't where it thought it was, it's going to say, well, I can't find this image. Can you point me to it again? So if you do that, just make sure that if you do need to move images around, just be aware that you will run into problems with that. I'm going now, image server down the bottom. The image server essentially is you can kind of think of it as the translation algorithm for the files. So every single file type has its own requirements to translate it to be opened. Now this one I'm pretty sure uses a default. I usually just default let QuPath decide. And I think QuPath literally just uses OpenSlide for most things, but you can use bioformats. ImageJ, I'm not entirely certain how well that would work, or the JSON server, but bioformats and OpenSlide should be fine. I typically just let QuPath decide. Auto-generate the pyramids should be on. That will make things move a lot quicker, especially if we're doing zooming. And everything else I typically leave off. Set image type, you can tell that. So if you have all HDAB images, you just say they're all HDAB, but this is actually a mixture of HDAB and fluorescence. So I'm going to leave that blank and I'm going to do that manually a bit later. So I'm going to click import. And now within my project, I've got a little window over the other side telling me that it's working. And then underneath my project, I now have all of my images. Now they don't have to all be the same. They can be just any image, any format, any staining that you want. And allow you to essentially, by double clicking on them, to bring that back up. Now, when I double click on it, it asks me, what is the file type? Now, it's pretty much worked it out itself that this is an HDAB, and I click apply, and now it knows it's an HDAB image. And we'll talk about color deconvolution in a bit, but it's done its automatic color deconvolution. So now if I click on this other image, it's going to ask, because I've made a change. I've told QuPath that this is an HDAB, and so it's asking me if it wants me to remember that. If I click no, it's just going to ask me for if it's HDAB again. I'm going to click yes, and it's going to open up this, and it's already picked that this is a fluorescent image, and I'm going to click apply, and now it's applied the requirements for analyzing a fluorescent image to QuPath. In the image tab over here on the left, you'll find the metadata for the image. Name, where it currently sits, the pixel type, it's a 32-bit float image. The magnification that it was imaged with was 10 times, the width, height, dimensions, most importantly, the pixel width and height. Now that's usually taken and stored based on the imaging instrument. So if you use a confocal, most confocals, practically every confocal these days will automatically assign a pixel height and width. And that is absolutely of requirements if we're doing any size measurements or if we're adding any scale bars. If you look down the bottom, you'll see there is a scale bar at the bottom left of the image. If I zoom down there is a scale bar there. So you can, and as I zoom in and out, you'll notice that that will change depending on the current zoom that I'm using. So it will always be correct no matter what. It's very dynamic. Let's put that back. Now, most of these can't be changed. Obviously, the name can't be changed because that's the name of the file. The URL can't be changed. None of this can be really changed. The only thing you can really change in here is the pixel width and height. If I double click on that, for example, if it's obviously wrong or it hasn't been recorded, you can then modify it. If you've got like stage micrometer data or some other data that you know what the pixel width and the height, sorry, a spam call, you can go away. You can change that pixel width and height, but practically everything else you can't do. Slide label, if you want to have a look, most slide scanners will scan the label of the slide. If you want to have a look at that, you can do that by going into view, and I can never find it because my eyes are terrible. Where is it? Show slide label right there. So if you click on slide label, now this one doesn't have one because it was obviously not taken with a slide scanner. But if I go back to the HDAB, and I go to view, slide label, there's the label for the actual slide. So if you want to have a look at sort of any details, then you can actually see that piece of tissue there on the slide. So that's been stored. Now, can I get rid of this? There we go. Navigation. Navigating can be done using the move tool. So you can either click this little directional button here or click M, and that will select it. And that allows you to use the left mouse button to move an X and Y, the wheel on the mouse to zoom in and out. You can also use this navigator up the top. Navigator shows the whole slide and where you're currently zoomed in on. So if I zoom in here, you'll see that little red box will get smaller. However, you can also navigate using this. You can actually pick this up and move it to where you need to go and zoom into specific areas. And you can also, I think you can double click around here and it'll automatically go, actually single click, it'll automatically go to where you want to do. Now, talking about the image pyramid, as I was talking about before. Now, as I'm zooming in, it's not actually digitally zooming in. It's not making the image that I'm currently looking at bigger or smaller. What it was actually doing is peeling off the layer above it. So I'm essentially going down through the layers of the pyramid to get to the full resolution of the image. We're not seeing anything else. We're just seeing this one part of it because the actual image itself is massive, but we're just sort of zooming in by peeling off those layers to get to the full resolution. So Fiji cannot do that because it has to open up the image all as one part, as one entire high resolution image, which is why it often falls over if I try to open this thing up. How big is this image? 26 gigabytes. So there's no way that Fiji could open this image up as one whole image. We'd probably have to open up the third or fourth layer of that image pyramid. Regions of interest. Up the top near the move, you'll find these shapes. Now, these are regions of interest to allow us to isolate specific areas of the tissue in order to analyze, to manipulate, to do whatever we want. So essentially what you can do is click on, say, the square tool, select this area like that, and then we can go in here and then analyze this or change a crop and do things like that, analyze that. Now, in order to change that, we need to click on the Move tab, and then you can grab it and move it around. I don't think it actually works if I do that. Yeah, so if you actually try to select the same tool, you can't actually change this anymore. All it does is try to draw another one. So what you have to do is choose the Move tool, select the annotation like that by clicking on, not inside, but on the actual line, and then grabbing these little points to change there. You can use the circle tool. So the circle tool is, obviously it's hard to draw a perfect circle. However, if you hold down Shift, you can draw a perfect circle as you draw. Now, one thing that is missing that I do like in Fiji is the ability to draw a circle from the center outwards. With QuPath, it will only draw, if you hold down, if you draw out like that, it will only ever draw out from the top. I remember with Fiji, if you hold down Control and Shift, it actually allows you to draw out from the center rather than the edge, but that's fine. You can just move it, then move it to where you want it to be by using the Move tool. Also available in here are, so if you draw a ROI, obviously it's clear in the middle. If you use the fill function, which is this, it will actually fill in the ROI to make it sort of look more obvious about the actual area that you're actually looking at. Clicking delete will get rid of it if you need to. The magic wand and the brush tools, which are up here, so the magic wand and brush tools are really, really quite helpful. So the brush tool is a large sort of ROI that allows you to essentially paint your ROI on. Now, annoyingly, it doesn't automatically fill it in. It just essentially, you have to then fill it out. I've selected off it. I think if I click that. Cool. All right. We have to select the annotation here. So your annotation list will store all of your annotations. And then I think I can then, yeah, and then you can paint within the annotation. So any new annotations that I build are automatically going to be added to the annotation list. Unlike the ROI manager in Fiji, where you have to actually manually do it, every single one is just added automatically. The other tool, the brush tool, the size of the brush will depend on your zoom level. So if I'm zoomed way out, I can draw a very, very large area with the paint tool. But if I'm zoomed in really, really high zoom, I can then use the paint tool to then do a very, very fine annotation. The other one which is really, really helpful is the magic wand tool. So if I select the magic wand tool, you can use that to select pixels of similar intensities. So if I click there in the space and then draw around, it will automatically find the margins of the area that I'm trying to select. So not just in the spaces, but also within the tissue itself. So it's really, really good at finding the margins within the tissue itself. So some useful keyboard shortcuts for QuPath. So M is move, B is brush, R is rectangle, O is circle, P is for polygon. So if you click around multiple points, you can create a polygon. W is for the magic wand. To show the ROIs all off or all on, you can click A. And to turn the fill on and off, it's Control F. It should be Control F, or is it F? Shift F, sorry, Shift F. I've got Control F there. Fix that. The annotation manager, which I was talking about before, is over here. If I zoom in over here, so there's your annotation list. So if you double click on the annotation, it will automatically select that area. So I'll actually come out. So if I double click on any of these, it will automatically select and center that particular area. If you right click on the annotation and click lock, you can't then change it. You can't modify the size. You can't add to it. It's now protected. You can delete it though. It can be deleted. The selection tool, which you'll find up here, that one, will allow you to use essentially just select parts of the annotation to stop. Is that right? Select, turn off selection mode with drawing tools into selection tools. So yeah, you can select these regions as you need to. I don't particularly find these really helpful as opposed to the ROIs. So you can use the selection tool if you need to. Classes. So down here next to annotations are the classes. So the classes allow us to change what the ROIs are actually highlighting. So if we're looking at, these are the defaults, which will automatically be installed. Now you can delete these, change these, add more, do whatever you want. So by default, it automatically defaults to tumor because a lot of the pathologists do tumor work. However, if you sort of found, okay, this area, so if I select this annotation here, is actually a necrotic region. So you can then right click and set the classification here to necrosis. And you'll notice that the color, if I deselect it, will change to the color that the necrosis is. Now you can change the color as required. So if you double click on it, you'll get a different color. So if you want to change necrosis to maybe a magenta, then you can do that as well. Excuse me. If you wanted to add a new class, you can go up here and you've got the plus and minus. So if you could click plus, you can add what's a function that I could do? Vessel. Vessel. No, I spell vessel right. Vessel. I think that's right. Okay, and then I get a new class that I can change the color to if I want to. Maybe I make that fluorescent green. So now I can right click and set that classification as I need to. So now that's selected as I need to. And of course you can just get rid of regions if you want. Remove, gone. Auto add classes. So if I go into here, you can, now it's not in here because auto add classes only work in the fluorescent. So if I select a fluorescent image, no, don't save. And I go into annotations, I can come in here. Now where is it? Select. I'm missing something. This is not a fluorescent image. Maybe if I go to this one. Fluorescent, apply. I go into here. Oh, it's missing. What I should have in there, and for whatever reason it's not there, is auto add classes. Auto set. No, that's not it. Yeah, I'm missing some functionality. So over here, what I'm supposed to have is populate from image channel. And I don't know why it's not letting me do that, which is very weird. They may have moved it somewhere else. All right, I won't linger on that. We're not going to be doing that with this one anyway, but before we do this, we'll use this image for machine learning. But yeah, before that, I will work out where that button has gone. Okay, anyway, so it is possible to, based on the name and the metadata of each of the channels, is to actually create all of the classes based on the channel names. For whatever reason, I can't find it in this, but I will figure that out for next time when we actually need it. Object hierarchy. Now, there is an object hierarchy to this, which you'll find in annotations. Now, the top of the hierarchy is the annotations. So if I draw a annotation, and then within the annotation, I then do some cell detections. So those are then the bottom of the hierarchy. So if we come over here, we can actually see the sort of the hierarchy that you'll get. So within the annotations, you'll get cells as part of those annotations. So the parent is the annotation and the children are the cells. So we'll get more into that when we look at cell detection. Workflow. Now, often when I'm doing image analysis at least, I have to try a whole bunch of different filters, a whole bunch of different processes in order to get the image prepared before I do my analysis. Often this involves doing filters and things like that, and trying to extract features. Now, trying to keep track of that can be very exhausting. So what they do have is a workflow indicator. So essentially all I've done so far is just set the image type. But when I start doing my, my imaging workflow, what it will do is it will remember all of the functionalities that I used in order to get the final result. So that allows you to keep track as a sort of a recording of what's actually going on. So I said that ImageJ cannot open these files because they're too big. Now, because QuPath was built around Fiji, Fiji has actually been built into QuPath, which you'll find the ImageJ button over there. So what you can do is if you want to do some work on a particular part of the tissue, you can actually come down here and say, and find a zoomed in spot and say, well, I want to do some analysis on here. I need to run it through ImageJ because QuPath doesn't have the functionalities that I want. So what I'd do is I'd select an area using my ROI, and I'd go into here and send region to ImageJ. Now, sending the region will take a crop of this essentially, send it off to ImageJ so I can do my whatever filters and things that I want to do, and then I can send it back to QuPath to then continue on with my work. You'll get this little window. The resolution I'm going to use for this, because it's such a small area, I can probably get away with the full resolution, so 1x essentially. But if you are using a very large area, you can actually say, well, I want to use maybe, I don't know, a quarter. So you'd use 4 instead. I'm going to use the full resolution. The resolution unit I'm going to use as microns. I'm not going to include this ROI because I don't need it. Include overlays. I'm not using any overlays. That's if you've already detected cells and things like that. Color transforms, I don't need to bother about that. That's just if you've changed the coloring. So then I click OK. Now, the first time you do this, it does take a little while. Oh, actually, that's a lot quicker this time. But once ImageJ is open within once ImageJ is open within QuPath, it should just essentially go straight in. So here, now I can use all of the plugins and things like that. Now, I've just installed this today, so I haven't actually set up my plugins. But if you have Fiji installed on your computer, you can actually redirect its internal ImageJ copy to use your plugins. So I'll show you how to do that in a second. But we can pretty much just use just standard ImageJ functionality. So if I want to do some color work, so I can use the channel tool to change it to a color. So then I can now go through my slider and see all of the different channels. I can use thresholding. So if I wanted to threshold that, I can threshold that, create a binary image, etc. Convert, blah, 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 convert to mask. And then what I can do is use the function in here. So send the send ROI, blah, 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 send overlay back to QuPath, send ROI to QuPath. So if I click that, it should send it back to, how did that work? Send ROI to QuPath. So, okay, for some reason it's not bouncing back. Overlay to QuPath. Oh, okay. It should be sending that image back to QuPath, and for whatever reason it's not. Strange. All right, we'll ignore that for now. We need to work out what's going on with that. Now, if your area is too large, it may pop up with an error saying, attempting to extract this region is likely to require XXX number of megabytes. Are you sure you want to continue? If it's less than a gigabyte, it should be fine. It should have no issues. If you're sort of getting into the several gigabytes, it'll probably fall over. So just be careful with that. Now, if we go into edit preferences and ImageJ, you can actually direct it to your ImageJ plugin data. So if I find wherever my Fiji is, and I can point it to my Fiji install and its plugins and select that folder. Now I'm going to close ImageJ down. And oh, did I save that? Yep, I did. Okay. So now what I can do is if I send this back to ImageJ again, see if we can open up. Okay, so now it's taking a little bit longer because it has to open my plugin. So now if you look at the plugin list, I now have all of my plugins, which I normally use for Fiji. Now, the one thing that I have found when I've been doing this that doesn't work is Stardust. Stardust is our AI nuclei detection algorithm, which we use in Fiji. But as of version 0.6 of this, it actually has Stardust installed. So you can actually use Stardust to detect nuclei within QuPath using Stardust rather than sending it off to Fiji. So the extension manager here, which you'll find in extensions. Extensions is like plugins, which we found for which we use in ImageJ or Fiji. So plugins you can add if somebody's written a new plugin. Oh, no problem, no problem, Michael. So with the extensions, we can add new plugins just like we would with Fiji. So if you download a QuPath, you can actually drop it into the extensions directory within the software and then install it into QuPath to use later. So I have actually installed Stardust. I had to do that separately, but that's pretty easy. You can pretty much just come in here and click plus. So I haven't installed these ones with pluses. So if I wanted to, the stitching extension, I could click that and install it like that. So I'm going to cancel that because it might make me restart QuPath, and I don't want to do that right now. So you can add new extensions fairly simply by dropping them into the extension. Oh, it's already done it. Okay, good. By dropping it into the extension file folder and then installing it. Right, part 3, channel navigation. Now, no matter what the image type, QuPath can handle multiple stain channels. Now, when we do fluorescence imaging with confocals and things like that, typically the fluorescent channels are housed in separate images. So each channel is imaged separately and is then overlaid to produce a multi-channel image. Now, when we stain with things like DAB and H&E, we typically take an image of the whole sample with a color camera. Now, there is no separation within the camera. It's literally just an image of the color, the pixels from the sample. Now, what we can do is what's called a color deconvolution, which allows us to separate those stains out computationally, and it's pretty accurate the way it does it because it's a very well-known stains, DAB and H&E. We've used them pretty specifically for many years. So those stains, what's called the stain vectors, are well known. We'll go over stain vectors in a minute. The brightness contrast command is a really easy way of visualizing the separation between the channels and navigating through the different combinations. So if we go back to QuPath, okay. So now we have this is a 7-channel image. So there are lots of different, this is a multiplexed image of a tissue sample, and each of these colors is a different type of cell based on immunohistochemistry. So what we can do is go up here to this little black and white icon here. This will open up the brightness and contrast function and will allow us to navigate through all these channels. So at the moment, all of them are turned on. So if I click that button, it turns them all off, and I can select each channel individually to show me where these different cell types actually are. So if I open up the DAPI, obviously DAPI is everywhere, so that gives me a good idea of where the tissue. Now these yellow are CD8 positive cells, so you can see where those actually lie within the tissue, and then you can select as many as you want on. Now, every time I select and highlight one of these, you'll notice that the histogram will change depending on the color. Now, with the histogram, I can use this to change the brightness and contrast of each of these channels individually, excuse me, by using these little sliders. So if I change the sliders, you'll notice that the intensity changes because I'm changing the range at which the display curve is displaying those particular pixels. So I can modify this on both ends to reduce the very low level pixels, or increase all the pixels to high intensities. So you can go through and optimize these for each of the channels. Be aware, this does not change the data itself. All we're doing is we're changing the way that it's displayed. So the intensity of each of these pixels is actually set, but the way that we see them is changed. So if we tried to do analysis on a cell that was this intense versus this intense, we'd essentially get the same answer because we're just changing the way that it's displayed. The channel viewer, if we use that, is a helpful way of separating the different channels in a single and seeing all of the channels in a single window. So if we go to view and show channel viewer, which I can never find, show channel viewer there up the top, that will give us a little window to allow us to navigate through. I'm just hovering over, I'm not clicking anywhere, but it actually shows me each of the individual channels as well as a combined channel view as well. So I can see at a glance what each channel is doing. So for a bright field image, it works a little bit differently. So if I double click on here and go back, I'm not going to save any of those changes. Come on, load up. There we go. Now, obviously this was taken with a transmitted microscope, wide field microscope with a digital color camera. This has been stained with DAB and hematoxylin, showing us the nuclei. The hematoxylin is showing us the nuclei, and the hematoxylin and the DAB is showing us where a specific protein of interest is actually localized. I don't know which protein this is. It's just a sample image that we can use. Now, as this was captured as a single image, we obviously cannot separate it based on the original data that was captured. It's not separated into individual channels. However, we can use an algorithm to do this for us. Now, this is done automatically. Whenever we open up the image, it's automatically done for us. So when I click on that, you can see that I'm selecting the original, showing me the original image. Now, if I click on hematoxylin, it will show me its computationally separated image. Now, if I wanted to, I can then use my my little sliders to change the way that this is actually. Now, sometimes you'll find that the actual this line is actually out of the screen, so you can actually just grab the little channel max tool and just use that to optimize the view so that you can see where everything is. And that makes it a lot easier to see where the nuclei is rather than what it was before. Now, if I click on DAB, it'll show me computationally where the DAB is actually located here. And again, I can use my little sliders to make this a little bit more obvious to see where that actually localizes. Now, this is done using what's called a stain vector. Now, stain vector is a red, green, blue value, which is used as essentially a characteristic of what the stain actually, the color of the stain. And it uses that with really good effect to separate these really, really well. So if we go into the image, we can actually see the stain vectors down here. So this is the metadata for the image. And we actually see the stain vectors here. So these are the red, green, and blue stain vectors. Brown is usually made up of mostly red and green with a little blue. Is mostly made up of green and blue with a little bit of red, I should say. Whereas hematoxylin is, actually, no, so it's the opposite. So the lower the value, the darker it is. That's right. So a lot of blue is in the hematoxylin with a little bit of red in there, and brown is usually made up with mostly red and green. Now you'll notice that there is a third channel, which is the residual channel. Now what the residual channel is are colored pixels, which the deconvolution algorithm could not assign to anything. It couldn't assign to hematoxylin, and it couldn't assign to DAB. So if we look at this, we'll see under residual, and there shouldn't be very much, but there's probably a bit. It looks very white, but if we change this, there is a bunch of stuff there. It's very, very low, and it's done a fairly good job of assigning most of it. We have to boost the signal quite a bit to see it, but most of the pixels have actually been assigned as red and green, as hematoxylin and DAB. So we can effectively ignore the residual. Now, if you find that your residual is much, much higher than the stains you're trying to deconvolve, you may have to manually change the stain vectors in order to suit the particular dyes that you're looking at. Because it doesn't have every dye in here. It has basically H&E and DAB, and that's pretty much it. If you're using something like Masson's Trichome or Picaceres Red or something else, you'll need to define that manually. Proceed with caution with that. However, it can go very, very badly if you haven't got the correct controls, or you choose the wrong area to go for the stain vectors. Yeah, just be careful with that. Now, to change the stain vectors, what you can just do is if you double click on the stain itself, you will get this stain vector. Now you can modify these values, but it's very, very hard to get right. These values have been just derived from a lot of work. So trying to do it yourself can be, like obviously HDAB is very well known. However, if you're doing something different, like as I said, Picaceres Red or Masson's Trichome, you can have a go with changing the values, but I do not recommend that. It's really hard to get right. It's much, much, much easier to use a control to take the stain vectors from that in order to change those values manually. Now, what I'm going to do is I'm going to find an area which is obviously hematoxylin positive. So maybe around, where's a good area? Yeah, so what I'm looking for is an area that's not contaminated with any of the brown DAB stain, which unfortunately is most of the place. All right, let's use this area. So there's your hematoxylin, I think. So what you can do is draw a small area, which is mostly blue. And then what you can do is then double click on the corresponding, oh, what happened there? There we go. Just crashed for a little while. If you double click on the corresponding value here, it'll ask you if you want to set the stain vector using this. Now, obviously, it's better to have a just hematoxylin and just DAB stain sample. That way you don't have the contamination here. So this would be a great area to choose because it's very heavy in nuclei, but there's obviously DAB here. So I'm going to click no. Now, if I cancel. So if I now go into maybe, where's a really brown area? Down here. And I find an area which doesn't have any hematoxylin, and I would select this, like that, and then double click on my dab. I can set my my dab stain vectors based on that. So I would only do that if I have to, if I'm using a strange stain. So if you wanted to, you can actually double click on the imaging type. So if it's not HDAB or H&E, you would click bright field other, and then you would set your stain types. Now, if I did that, I can then modify the name. So if you've selected HDAB and H&E, you can't change the name, but if you have changed to the other stains, you can say that this is Masson's trichome, whatever it is, and then set your stain values based on that. But you can't do that within the HDAB because it's set in stone. Now, the QuPath can use Z as Z images and time images. So that's new to version 0.6.0. Previously, it wasn't really. You could do it, but it just didn't work very well. Apparently, this one is much, much better. You will get some sliders up the top here. This is for channels, but you will get one for Z and time that you can then cycle through. But I don't recommend doing it here. There's other software that you can use, but I suppose if you're stuck, it's not the worst thing. I haven't actually used it very much for time because I mean, obviously, why would you ever use a time image for pathology slides? Z, on the other hand, I can see some merits for doing that if you've done what's the word I'm looking for? Serial sectioning. That would work really, really well. Right, so that is all about channels, and very important to know how to do channels with QuPath. So we're getting into the analysis part of it. Now, this is more simple than what we will be doing in part 2, which will be the machine learning part, but this will allow you to use QuPath's algorithms to detect cells and measure them, as well as find whether a cell is positive or negative for some particular marker. So manual measurements, you can do them pretty easily using the ROIs. Just like you would in Fiji, you would essentially just come into here, zoom in. So if I want to, say, measure this piece of tissue here, I'd use maybe a paintbrush or maybe magic wand tool. Let's use the magic wand tool. See how we do. Let's select this area here. And then what I can do is go into measure and show annotation measurements. And then that will show me all of the annotation measurements that we have. It'll show you a little picture of what each annotation objects looks like. So it's very easy to figure out which one is yours. And then you go across here. It's very basic measurements, such as area, perimeter, the centroid, the X and Y centroid, whereabouts in the image, the center of this particular image is. Nothing on intensity or things like that, but you can get the size of that very easily. From that, you can save this data as an Excel results data table for use for your analysis. So very, very simple to actually do that. Also, if you wanted to do, so I'll just get rid of all my annotations, and delete, gone. Now, let's just say I wanted to start counting nuclei. I'm going to turn off the DAB channel, just use hematoxylin. Using the little counting tool here, I can just essentially go through and manually select these, and you see a little spot here. So if you want to bypass more sort of technical analysis and you just want to, manual analysis is fine. It's more accurate, specifically it is more accurate than doing machine learning or anything like that. But just remember, your time is very precious. I'm a 95% correct person. If the computer can get it right 95% more or less, that's good enough for me, because you've got to think about how long this is actually going to take. If I want to do every single nuclei in here, it's going to take me hours, if not days. So I'm not going to do that. But if you are doing a small area, often it can be just easier just to do a quick count with that, and it will keep a count of the analysis. Now I can change my can I do that? My class, color. So I can change the color of the different classes that I want to use. So maybe this is a different subtype of nuclei, and you just add different types in here. You can call them whatever you want. So type 1, apply, and that will keep track of all those. Now, not just time, but fatigue. If you've ever done some long-term manual analysis, eventually you get tired and you get lazy. So things just tend to get a bit sloppy. So manual analysis is fine, but if it's taking a long time, then you should really look at computer-assisted analysis, which is what we're going to do. So manual versus automated analysis. So manual analysis can often be easier than attempting to create an automatic workflow. And it's fine if you've got a few images, or if the object is far too difficult to detect via software. Sometimes you get an image which the software just would not have a hope in hell of actually being able to do anything with it. If you've got lots of, like if you've got an overly confluent cell, images of cells, or things are just not well resolved enough for the computer to actually do anything. The human eye is far more sensitive. However, if you can't determine if one object is 2 objects or 3 objects, if they're too clustered, then the computer will have absolutely no hope. However, if you have several hundred images and the object you are trying to measure is detectable via software, then it can be advantageous to put the time into creating an automated workflow. So there are 3 separate functions which we'll be going over in these workshops. First of all is the automatic cell detection, which we'll go over in this workshop. And then in part 2, in probably October, we'll go over the object classifier and the pixel classifier, which uses machine learning to detect specific objects which we're looking for. So cell detection. Now, this allows us to automatically detect, count, and analyze cells in an image which have a nuclei marker. So you need something, specifically a nuclei marker, to tell where a cell is. Now, just be aware that when we're detecting cells, we're mostly detecting the nuclei. These cell margins, they're added automatically based on the shape. You can actually see the shape is just an enlarged version of the nuclei. It's not real. It's not based on the tissue below it, because there is no signal for the tissue below it in these images typically. It's literally just an expanded part of the nuclei. So you can see it very obviously here with the fluorescent detection. So it's obviously detecting the nuclei, but then it's not used. This is very, very obvious that this is the cell margin. However, it's just literally just expanded out from the nuclei. So it's based on what we tell it. We essentially say, expand this far out from the nuclei. And so you can't actually do any analysis that way. However, you can do cell counts, and from that you can do detection of another marker, for example, DAB, in that general location. So just be very aware of that. Okay, so let's go over the cell detection algorithm. So you'll find cell detection in analyze cell detection and cell detection. And that gives you this little window here for using it for changing your settings. Right, the detection image is obviously going to be hematoxylin because we've got a hematoxylin image here. You can use the optical density. We'll talk about optical density in a bit, but very, very generally, it's how dark the sample is. The higher the optical density, the less light can transmit through, hence it's more dark. But most of the time, we'll just use the optical density of the hematoxylin rather than the optical density of the cell in general. The requested pixel size. Now, what we can do is by using a higher pixel size, we can reduce the resolution of the image, which makes it faster, especially if we're doing large images. So we can reduce the resolution artificially of the image. Now, the actual pixel size in here, if I go over to images, is the actual pixel size is 200 microns. So essentially, we're doubling times 4. Yes, effectively times 4 the pixel size. Even though it's times 2, remember it's an X and Y. So you by reducing that resolution, you speed up the whole thing. Now, by doing that, it can make it go faster, but you do lose resolution of the image, which can make things essentially blur into one rather than 2 separate nuclei. So just be very careful with that. The background radius is a filter. That should be roughly the radius of the nuclei. So nuclei are usually between 8 and 10 microns. Essentially, it filters out any objects below that. Use opening by reconstruction. I had that down and now I can't remember what it is. That's weird. Ah, there it is. User opening by reconstruction. What that does is it improves the accuracy of cell detection with uneven or complex backgrounds. So it helps refine the background estimation by performing an opening operation followed by a reconstruction, whatever that means. It's just a workflow that they use. It can produce some artifacts, but I typically have it on by default because it does make the whole thing a lot more accurate. The median filter size and the sigma filter size, those essentially blur the data. And I know with images, blurring the data seems like counterproductive because it makes the image look terrible. However, when we're doing object detection, and I use this all the time, if you've ever had a script written by me, you'll often involve some sort of object detection. You'll usually find me using a Gaussian blur rather than a median filter or sigma filter. But what that effectively does is if you look at some of these nuclei, it's not one big blob. It's actually a lot of sort of, there's structure in there. So what the sigma filter and the median filter do is blur that into more of a blob. So by doing that, it aids in the detection of this as a solid object. So I don't usually use the median filter. I usually have that set to 0, but the sigma filter I found works really well. Depending on you can increase and decrease both of them, and try different things if you're having trouble to try and improve that. The minimum and maximum area is essentially filtering out objects which are that can contaminate the results. I know we've done that here, but this is just an extra filter to allow us to get rid of large clusters of objects and very, very small sort of things. So these work in tandem to reduce debris essentially. The intensity parameters, the threshold is the threshold is essentially an intensity value, below which things are ignored. So this is usually set pretty low because the samples, when we're dealing with fluorescent, it's usually bright. However, we're dealing with stains, which block light, essentially, which makes it darker. So these thresholds are usually quite dark. If I put this to 2 or 3, it probably wouldn't work very well. So 0.1 or even 0.05 is usually where I sort of play with the threshold. The max background intensity is really only useful if you've got tissue like this with folds in it. So here, this is problematic because there's a lot of garbage there. Now, if it's trying to do a detection based on the darkness, I suppose, of the tissue, this is fairly dark. So this actually might be detected as an object. But what it actually does is it just ignores this flat out by using that. So you can change that to ignore this. Split by shape, if you've got objects, which you often do, nuclei aren't usually too much of a problem because they are typically separated by the cell membrane. But what it can do is, if you have objects which are very close together, is it splits by shape. So you can use that function there. It takes a little bit extra time, but well worth it. Exclude dab. So what it does is it just excludes dab. If it's excludes dab in the membrane. So if there's any dab in the membrane around the outside, it just ignores that flat out. Cell expansion. I usually have that turned off, by the way, because I want to see all of the dab. Cell expansion. Now that's what I was talking about with the cell membrane, the cell margin. This is where we set that value. So if you know your cells are typically, I don't know, 20 microns across, and the nuclei is 10 microns, which would mean we would want the cell to expand 5 microns out from each side. So you've got 10 microns across here, plus 5 is 20. So that's your cell expansion. Now you can make this as big or as little as you want, but it's just up to you what you actually want to do. Include the cell nucleus. We usually have that ticked off unless we just want to see the membrane, the margin of the cell. So it skips showing us the actual where the nuclei has been detected. You can turn that on or off elsewhere, and I'll show you where that actually is. Smooth boundaries just makes things look smooth. So if I come back here, smooth boundaries essentially turns it from this to this. Essentially, it might make things a bit quicker. I don't think so, but it just makes it look a bit nicer. So you can use smooth boundaries, and then finally make measurements. What we'll do is it will actually measure each and every one of those cells, and typically that's what we're trying to do. So we typically make measurements off. If we're just after a pretty picture with showing us where the cells are actually detected, we can turn that off. But I don't see any point having it off. You don't have to look at the measurements if you don't want. So what I'm going to do is I'm going to do a measurement over here. Let's go in here where there's some actual nuclei. Now, you cannot do this on an entire image. If I try to do this, it's going to fail. It needs, as I was talking before, you need, about the hierarchy, you need a parent for the children to exist in. So the children are the cells, and the parent will be the annotation. So what you need to do is come in, find your area that you actually want to analyze. That looks like a good one. And just draw a box. Big, small, doesn't matter. The bigger it is, the more time it's going to take to analyze. So I'm just going to draw a small-ish box. That should be a fair few hundred nuclei. And then I'm going to click Run. And off he goes. So the bigger my area, the longer this is going to take. It shouldn't take too long. That's a fairly small area. And there we go. Now, there are all my cells. And if I zoom in, I can see that it has detected some cells, but not all cells. So you can see that the more obvious, brighter ones can be seen there with the cell margin around. So what we're going to do now is we're going to try and improve this by changing the settings. Now, the most likely one I'm going to use is probably the threshold. I'm going to reduce that a little bit more to see if I can get that to run. Now, what I might actually do is I'm going to make that a smaller area just because it's actually thousands of cells. It's 4,000 cells. I'm actually going to get that. It's going to ask me if I want to keep the descendants. No, I'm going to delete those as well. I'm actually going to go for a smaller area just to make it go a bit faster. So let's drop that threshold to 0.005 and run that again. And that's much, much better. Much, much quicker as well. So I do suggest you start off with a small area just to test the settings. And then once you're happy with that, you can expand that to a larger area. Now, as I said, I'm a 95% person. As long as I've got 95%. Now, down here, we've got a few issues here. That cluster of cells has obviously had a problem. We can try and fix that by reducing the blurring filter. So maybe if I make 1 micron instead of 1.5, that will allow that to be separated a little. There we go. So now we've got a bit more separation. And I can continue playing with this as well. What else can I do? Requested pixel size. Maybe if I change the requested pixel size to a bit smaller, so 0.2, it'll give it a bit more resolution in order to determine that. Now, I think the reason I've got such crazy cell types now, cell sizes, because I've reduced that sigma filter. So what that sigma filter is doing is blurring it into a blob. So I think by doing that, I've upset the rest of it. Now I've still got this problem. Now, in the grand scheme of things, I mean, there's sort of a few issues around here where clusters of cells are running into problems, and you could sit here changing these settings until the cow comes home. But sometimes it's just not going to work because they are just, the software just cannot find the difference between these cells. So I might change this. Maybe I'll go down to 0.2 and see if I can improve that detection a little bit better. Still not perfect. What else can I try? Intensity is probably not going to help me. Oh, split by shape. I had that turned off for some reason. That's better. Yeah, I don't know why I had that split by shape turned off. Thought I had it turned on. But anyway, that seems to be a little bit better. I might change the threshold back a little bit more. Oh, too big. So it didn't detect anything because my threshold was too high, and essentially all of the nuclei were below 0.5. So 0.1 seems to be the winner. I'm pretty happy with that. That's probably about as good as I can get with this particular image. It's not too bad. So then if I wanted to, I can then expand that out to a much, much larger area. Now, if I go over to annotations now, I will find another tab called hierarchy. And under the hierarchy of this particular annotation are all the cells which were, so if I go in here and double click on each one, it'll actually show me the cell. What's that? It'll actually show me the cell that was actually detected. And that's pretty good. So now what I can do is go into show detection measurements. And now for every single one of these cells, I now have a measurement for how big they are. I've got a bunch of stuff actually. So rather than that sort of simple annotation, I have a lot of different values. Now, what it's done also is it's not just analyzed the nuclei, but it's also analyzed the hematoxylin, which is still it's still there. We just weren't showing it. So what it's done is it's taken a measurement of each one of these regions and produced some measurements. So if we look over here at this one cell here, we can go in here and find where is it? There's the DAB. So these are all hematoxylin. Here we go. So nucleus DAB OD measurements. So that's the optical density. So the higher this, the lower this number, the more obviously brown it is. The lower that number, the higher the number, the more brown it is. So if I find the brownest one, which is up here, there we go. So that's a pretty brown one. So that's your high expression of DAB. So it's done that analysis based on the intensity that it measured within this thing. So that's based on the optical density. And then from there, of course, you can then save your data and export that to your Excel for analysis. So if I find the lowest one there, what was it? It's DAB. So what it's done is it's done a lot of things. So we've got the amount of DAB in the nucleus, the amount of DAB in the cytoplasm, or what it thinks is the cytoplasm. So cytoplasm, DAB, and do we have it as a, yeah, and then the cell. So we've got 3 separate values over and over again. One for the a bunch for the nuclei, a bunch for the cytoplasm, and a bunch for the cell. So if we looked at the hematoxylin OD mean for the cell there, then this should be the most dark one, and that one should be the dimmest one. That's not right. Hematoxylin cell. Hematoxylin. Sorry, I want DAB. What am I doing? Cell DAB OD mean. Cell DAB OD mean. So that will be that one. So if I rank it based on that one, then that should be the least amount of DAB. And then this one should be the most amount of DAB, which is a very, very dark cell. So what we're going to do now is we're going to move on to a very, very similar function, but actually ranking these cells colorimetrically via the amount of DAB detected within it. So we can essentially mark each one of these cells based on the amount of stain that is actually found in it. For example, DAB, which we're going to do, eosin, and Mass's trichome, Picocereus red, whatever you want to do. Now, you do need to make sure you have the correct stain vectors set up for this. So you'll need to make sure. So DAB and H&E is obviously simple, but if you are using one of the other stains, you will need to work that out using the workflow that I was showing you previously. So what we're going to do is we're going to go to, instead of going to analyze cell detection, we're going to go down one to positive cell detection. And you'll notice that this looks very, very similar. It's got the exact same options that we had before, but down below, we've got this other function, which allows us to set a threshold for what we think is a positive and what we think is a negative cell. So what I'm going to do is I'm just going to essentially select another annotation over here. Maybe here. So we've got a lot of low intensity and a lot of high intensity. And I'm just going to draw a little box and check my settings. I think these should be more or less what I had before. And I'm just going to go and run. Now, we have a result here. Now, the red ones are DAB positive and the blue are DAB negative. Now, the DAB, whether it's a DAB positive or DAB negative, is derived from these thresholds here. Now, I have single thresholds set up, which means I'm only using this slider here from 1.5, which is the maximum optical density you can get, to 0, which is the minimum. So that would be a very, very negative cell. So what I need to do now is I need to work out what the optical density of a positive cell actually is. So I'm going to have a look around and find what I think is probably the lowest value that I would expect for optical density, and probably maybe this one here. So it's got some brown, it's being marked as negative. So I'm actually going to select that, and I'm going to have a look over here at the measurements that it's actually made. And what I'm going to do is I'm going to come down here and I'm going to find the cell DAB OD mean, which is 0.164. So what I'm going to do is I'm going to take that value and I'm going to push this down so that the value for the threshold is now 0.16. So anything over 0.16 should be now a positive. So if I click run now, this should go from a blue cell to a red cell, hopefully. No, why didn't that work? Dab. Oh, okay. I know what I've done. So I've set it here to nucleus DAB OD mean, but I've actually taken the nucleus, the cell OD mean. So what I need to do is come down here and say cell DAB OD mean. So that's why that failed. Okay. So let's turn that off and now run that again. And hopefully this time, there we go. So now we've changed the negative into a positive. So now I've got this threshold. Now you can optimize that however you want, depending on what you think. It's very subjective, of course, but based on obviously an educated guess about what is a positive and negative. But if you're sort of looking around here and say, oh, actually that's a negative, maybe that should be a positive. So I can go in here and say, okay, OD, so cell OD mean for DAB is 0.1582. So maybe if I make this 0.15 and run it again, that should turn from a positive. Oh, hold on. I need to turn off. So I'm selecting this, but I need to select the actual annotation. Run that there. And now that's a positive. Sorry, a negative. Actually, sorry, it's a positive for DAB. So that's how you would do that optimization. Now, you'll notice that there are actually 3 sliders. So what you can actually do is set up different thresholds. So with this, you can set up 4 separate categories. Below threshold number 1 down here would be DAB negative. From threshold 1, this value, to threshold 2 would be low DAB. From this value here to this value here would be medium value. And then anything above here would be high value, would be high level DAB expression. So again, you would need to do this manually. You'd need to go and work out what is actually a moderate DAB. So you may come in here and say, what would I consider a moderate DAB? Maybe that one. So that would be moderate DAB. So let's go to here and work this out and say, okay, from there to cell DAB mean, maybe 0.34. Maybe 0.35. Then I need to figure out what is the brightest, what is the most sort of intense that I would expect. So maybe that one. And you do that again. OD mean. And of course, you could use any of these values. If you're only looking at DAB in the nuclei, then of course you'd only score it's called score compartment. Wherever you're trying to measure that, you would just do it in the nuclei. But I'm going to change this to 0.7. So anything over 0.7 is now high DAB. So now if I run this again, I need to select my annotation and run. Forgot to turn off single threshold. Turn off single threshold, and now it's going to take all 3 of these into account. And now we get a whole bunch of different colors. So obviously, blue is negative, yellow is moderate, orange is, blue is none or low, yellow is lowish DAB, orange is moderate DAB, and red is high DAB. So that allows you to rank this based on those thresholds there. So then from that point, you can go into your measure, show, detection measurements, and then you would get your measurements based on that again. So you can see the different colors based on the intensities in the little snapshot. This just shows you what optical density actually is. What you would work out is the amount of light transmitted through a sample, and it's the transmission, and the optical density is equal to the negative log 10 of that transmission. So the higher that value, the less light has been transmitted. So you can get 1.52 transmission, absorbance through, which will give you the higher values in the amount of DAB. No problem, Mishka. So then we would have our results, which we've just shown you. So show there, you can export them to Excel for analysis. And that's it. That's how to use QuPath, and we're 20 minutes early. Fantastic. Good to know. So coming in mid-October will be part 2. So we'll go over a quick refresher of the basic functions, and then we're going to move straight on to analysis via machine learning. So object classifier would be very similar to what we've been doing with the cell detection. So detecting cells, determining whether they are positive and negative by training it, rather than setting a value like we have been for cell detection, just like choosing values like this. What we're going to be doing is we're going to select, we're going to train it using a individual channels. So it learns what is positive and what negative is based on that, and then apply that to an actual multiplex image, the multiplex image that we've been looking at, the fluorescent one before. Pixel classifier is more for sort of detecting things. So if I wanted to, I did this a while ago. I had an image I saw on the news of, I think it was about 200 flamingos, and I trained QuPath to detect what a flamingo was and what isn't. So that would be the pixel classifier. So we can use it. We can train a particular image to look for a specific thing. So we'll do that in October. So thank you everyone for attending and making it all the way to the end. But yeah, obviously a little bit more bite-sized than 4 hours. I prefer an hour and 40 minutes. So yeah, I'll send you out a calendar invite closer to the time if you want to come to part 2. So thank you very much.